Okay, I think we might get started. So, good morning, everyone, and welcome to the first of a series of webinars that are presented by members of the Industrial Transformation Research Hub in biocatalysis for pharma. So my name, as you probably all know by now, is Elizabeth Gillam, and I'll be presenting the first of this series of webinars. I should actually switch to the previous slide so that you can see a little bit about the hub. So the emphasis in this series of webinars is going to be on presenting some of the science that's led to the formation of this research hub. So we're going to be covering a lot of different aspects of research relevant to biocatalysis. Through this research hub, we hope to achieve a step change in the implementation of enzymes as biocatalysts in pharma. Through this series, we're going to be introducing you to some of the members of the hub and demonstrating the expertise that they bring to the hub, as well as illustrating some of the projects that we could pursue in the hub. So this is our schedule of talks. I believe everyone should have received the schedule already, but to make it easier to identify what aspects of the consortium we're going to be covering in each of the talks, we have a little bit of a color coding system here that relates back to the logo for the hub that I showed on the last slide. So we intend that these webinars may become a forum for discussion of the priorities that we need to address in order that enzymes can be used to their full potential in chemical processes in pharma. And we also hope that these will serve as a bit of a model for what to expect from the regular forums that we're going to have in the hub that we're going to use to communicate outcomes and to ensure that participants have timely access to information and tools resulting from the research underway in the hub. Today, I'm going to be talking about a platform technology on which this hub is really based. In other words, the ability to stabilize enzymes so they're robust to process conditions, such as long operational times and elevated temperatures at which they would be used in industry. So for a number of years, my lab's been working on cytochrome P450 enzymes, and you'll probably all know that these are one of the most versatile and powerful types of enzymes, which can catalyze a diverse range of reactions that are relevant to industry, potentially with a degree of chemo, regio, and stereoselectivity that's difficult or impossible to achieve using pure chemocatalysis. So P450s are potentially useful at many stages of the drug discovery and development pipeline, from identifying the sites of metabolism at the start, and generating analogues for structure-activity relationship analysis, to producing metabolites for structural identification and toxicity testing, and in principle, for large-scale synthesis of drug candidates. We can also envisage them as being used for bioremediation to clean up the waste stream from the drug pipeline, a stage that doesn't usually feature in diagrams such as this, but is increasingly important considering the requirement for more sustainable manufacturing processes. However, we all know that natural enzymes are not really well suited to industrial conditions. So we hope that we can use enzymes at any stages of the drug development pipeline that need improved chemo, regio, stereo selectivity. And we can also hopefully introduce them to produce more environmentally friendly processes using less solvent, and to treat the waste stream, treat potentially toxic byproducts prior to release, so to have less environmental impact from the chemistry done in pharma. But enzymes are not well suited naturally to process conditions, and some of the things that we need to address are really shown here. So thermostability, the ability to use enzymes at elevated temperatures, but also for longer times is important. Solvent tolerance, we often want to load up reactions with as much substrate as possible to increase the amount of substrate available to the enzyme and therefore the product yield. Enzyme production costs are very important to consider because the enzyme is often the most costly part of a reaction, a biocatalytic reaction. So we need to minimize the costs. And most of the enzymes that we've been using to date are produced using rich medium, which is quite expensive. Stability to oxidative conditions is important because many of the enzymes we're going to be talking about generate reactive oxygen species when they're not tightly coupled. Substrate and reaction selectivity is, of course, important. We want the enzyme to be as selective as possible and to have as high catalytic efficiency as possible towards production of the correct metabolite or the desired metabolite. And with enzymes such as P450s, we need to think about cofactor supply. So NADPH, the natural cofactor, is expensive, and it would be ideal to be able to substitute that for a cheaper alternative. And finally, product isolation is often a very expensive part of a process to generate a metabolite or, for example, a drug candidate. Recovering the product from a reaction can often be quite problematic and low yield. So my lab's been trying to address these points over the last roughly 20 years by protein engineering, by different methods. But about 10 years ago, we started using a method called ancestral sequence reconstruction to engineer enzymes. So just to let you know a little bit about this method. So this is a bioinformatic method. It involves collecting the sequences of all of the known forms of a particular protein family, such as the cytochrome P450s. The first ASR we did was on the CYP3 enzymes, which includes CYP3A4 and 3A5 and 3A7, human P450s involved in drug metabolism. So we collected all of the sequences known for the CYP3 enzymes. We used some phylogenetic techniques to align them and to produce an evolutionary tree or phylogenetic tree, and combined these with an evolutionary model and a tool for inferring the ancestors of this phylogenetic family. And we inferred the oldest ancestor of the whole CYP3 family, and we call this N1, node 1 of the evolutionary tree. When we did this and resurrected the protein, so synthesized the gene that corresponded to the protein and characterized it, we were quite surprised to see that it was extremely thermostable. So here we show a result that shows the amount of P450 left intact, after an incubation of 60 minutes at each of the temperatures here. So you can see that the extant forms, including 3A4 and 3A5, all really fell apart at around about 37 degrees, whereas the ancestral form was stable to about 30 degrees higher temperatures. And we can see a value here that 60 T60 represents the temperature at which 50% of the protein is still intact, still functional, after an incubation of 60 minutes. So this was quite a striking result. This means that the enzyme is stable to higher temperatures, but it also means it's more long-lived at lower temperatures, at ambient temperatures, for example. So if we compare the half-life of the enzymes at different temperatures shown here, so 50 to 75, if we take 50, for example, compare this ancestor CYP3N1 to the two major human P453A forms, we can see that at 50 degrees, the extant forms, the human forms, fall apart in a couple of minutes. However, the ancestral form lasts for about 10 hours. So this is quite a profound stabilization of the enzyme. And this also applies at room temperature. When we purify these enzymes, we can leave them on the bench at room temperature for months, and they basically don't suffer any deterioration. This also translates to a higher optimal activity, optimal temperature of activity. So this shows CYP3A4 in blue, CYP3N1, that ancestor, in orange, and you can see that the optimal temperature for activity is shifted about 20 degrees. So this is a very effective way of making enzymes thermostable. If we compare this to other methods that have been used for stabilizing enzymes, such as methods in the Arnold, Halpert, Vermeulen, and Commandeur labs, some of the results with P450BM3, CYP102, are shown on the left, and some of the mammalian CYP2B enzymes are shown on the right. So you can see here that there's been a number of different methods applied to stabilizing P450BM3. We start off at a quite reasonable 10T50. So here we're comparing 10T50, which is a parameter that most people use to compare enzymes. So here we've got a 10T50 of the native enzyme in the 40s. And through a number of different steps, many different studies, there's been an elevation in the 10T50, which has been quite significant, but it's taken a lot of work. There's a couple of decades of work summarised here. For us, in one step, we've been able to achieve a higher thermostability, and that was starting off a lower base. So the extant CYP3 enzymes are less stable than P450BM3. So we think this is a very powerful technique. And I'm showing results for P450s, but I should say that we've now applied this to other enzymes, and it seems like a general phenomenon that we can increase the thermostability of these enzymes by inferring the ancestors. So we wanted to prove whether this was just a one-off. We redid the CYP3 reconstruction using many more sequences. And we wanted as well to infer some of the intermediate ancestors here. So this is the second version of this reconstruction. And you can see the nodes of the ancestral, of the evolutionary tree that we reconstructed. And here we can see that those intermediates in that evolutionary tree have intermediate thermostabilities as well. But they're all still quite significantly elevated compared to the extant forms. When we look at different P450 families, this is a reconstruction of the tetrapod clade of the CYP2 family. So this includes many of the drug metabolizing P450 subfamilies that will be familiar, 2Cs, 2A, 2B, 2E. And you can see here, as we go back in time, this x-axis here is relative branch length. So that can be taken as a surrogate for evolutionary time. So as we go back in time, we go up in thermostability. So the older ancestors are typically more thermostable than the younger ancestors and in the extant forms, which are at the end of each of these lines. So these lines connect ancestors that are related in an evolutionary lineage. But we can see that we've got two different broad groupings here. One of these lineages is much more thermostable than the other. So we can have events that stabilize particular lineages. So some are naturally more stable than others. So we've applied this now to many different P450 families, CYP1s, the CYP2D, which are earlier branching than the CYP2 clade I just talked about. This was a clade that I just talked about, the CYP2 tetrapod clade, the CYP3s and CYP4s. Quite confusingly, sorry, I've shown this as younger to older rather than older to younger. But you can see still that there's this trend towards increased thermostability in the older ancestors. And you can see that we've got now quite a number of more or less thermostable P450 forms. But these aren't just thermostable. These are also stable to solvent. So this is some results from CYP3N1, that original ancestor of the CYP3 family, comparing it to CYP3A4. So 3N1 is here in blue, 3A4 is in gold. And you can see that with methanol and acetonitrile at 37 and 50 degrees, we have a shift in the in the solvent tolerance, such that the ancestor is more solvent tolerant than the extant form. And we can have a look at this again with those intermediate ancestors and see that there's a correlation between thermostability and solvent tolerance. So the more thermostable, the more solvent tolerant these ancestors are. And this gives some of the 50% tolerances for each of those solvents. So you can see that these are quite substantial. But it doesn't end there. We can also express these enzymes much more efficiently. They have higher expression levels generally than the extant forms. So this was a reconstruction of the CYP2D subfamily of drug metabolizing P450s. And normally if we were expressing an extant P450, one of the human P450s in bacteria, all our expression is done in bacteria, we'd be very happy to get about 200 to 300 nanomoles per litre of an extant P450 in E. coli. Here we've got at least an order of magnitude improvement in those levels of expression. So you can see that some of these are going really quite high up to the micromolar concentrations in a flask culture. And this is enough to, for example, purify enzyme for crystallization. So these are crystals of 3 different CYP2D ancestors here. So we can start to solve the structure of these enzymes, which is really useful. We can start to work out why they're thermostable. We've just started to look at these enzymes in fermenter cultures. So typically we express in flask cultures, but we've just started a few trials in fermenter cultures in collaboration with some colleagues at UQ. And with this tetrapod ancestor that was expressed at about 7 micromolar in flask culture, we can increase that around about 4-fold by going into flask culture. So we're getting up to really quite high concentrations of enzyme. So this is increasing the yield of enzyme and making it cheaper to obtain these enzymes. We can make it even more cheap by using chemically defined medium. So this is comparing 3 different types of medium: minimal medium, a chemically defined medium, the contents of which I won't go into, and terrific broth, which is the typical rich medium that P450s are typically expressed in bacteria. And you can see that while the terrific broth, the rich medium, is still the best, we're getting very substantial levels of expression in the chemically defined media. And we can also express at 37 degrees, whereas normally we have to express P450s at 25 degrees, otherwise they just end up misfolded. But what about their activity? So this is a comparison between CYP3A4 in gold and CYP3N1 in blue. Here we're relating all of the activities towards a lot of different substrates. You'll know that CYP3A4 is a very, very promiscuous enzyme. It metabolizes a lot of different chemical structures, so a lot of chemical diversity in its substrate range. Here we've normalized the activity of CYP3A4 to 100%. This is a log scale. And you can see by the shadow of the CYP3N1 activity that CYP3N1, the ancestor, is more or less metabolizing the same range of substrates. Sometimes it's a bit better, sometimes it's not as good as 3A4, but it's effectively having the same activity towards this range of substrates. When we drill down a little bit further and have a look at the catalytic efficiency of these enzymes, this is looking at CYP3A4 and 3A5, the human, major human forms, with all of these other CYP3 ancestors, the intermediate ancestors, as well as 3N1. This is looking at testosterone 6-beta hydroxylation, a classic reaction for the CYP3 enzymes. And you can see that in blue, we've got 3A4, the best of the extant forms, but there's a form here, CYP3N19, that's much better, much more efficient than 3A4 at this reaction. We can also see a variety of different regioselectivities. So even though 6-beta hydroxytestosterone is the major product from testosterone from all of these enzymes, with some of these forms, we're getting significant amounts of other minor metabolites coming through. So the regioselectivity can be quite different. We can also find novel metabolites. So when we looked at midazolam metabolism, we can see that we don't just get the 1 prime and the 4-hydroxy metabolites that we always see with 3A4 and 3A5. We also have this third metabolite. So we can get novel reactions with these ancestors. This gives an indication of some of the regioselectivity differences we can get in between the ancestors. This is testosterone again. There's a number of different sites on testosterone that can be attacked by P450s. And here we're summarizing the results from screening all of our ancestors that are in bold, as well as the corresponding extant forms in normal text. And you can see that different ancestors will hit different parts of this molecule. So if you're considering testosterone as a drug-like molecule, in our library, we can find an enzyme that will metabolize any given spot on testosterone as its major metabolite. With P450s, though, there's sometimes a problem related to their promiscuity, to the fact that they're not highly specialized to any one substrate. And that is that if the catalytic cycle is uncoupled, they can generate reactive oxygen species. And we think that the generation of hydrogen peroxide, particularly, might be the thing that limits their activity on long-term incubations. So this shows a very cartoon representation of this P450 catalytic cycle. So it starts here. There's a couple of single electron transfers here. Oxygen binds here. So we're basically reducing oxygen. So we can get this cycle short-circuiting, either here to generate superoxide or here to generate hydrogen peroxide. And this happens particularly when the fit between the active site of the enzyme and the substrate isn't optimal and there's water getting into the active site. So we wanted to see whether our ancestral enzymes were resistant to damage from hydrogen peroxide. And we were quite surprised, pleasantly surprised to find that ancestral sequence reconstruction, at least in one case, seems to protect against oxidative damage. So this wasn't a drug metabolizing P450. It was CYP11A, which is a steroid biosynthetic P450. We did a reconstruction with some colleagues in Germany and resurrected the oldest ancestor as well as the mammalian ancestor of the CYP11s, or CYP11A, I should say. When we did this, we looked first of all at thermostability, of course, and you can see here the oldest ancestor showed the expected shift in thermostability compared to both the extant form, which is a bovine form in this case, and the mammalian ancestor. This is looking at a P450 spectrum. So this is the typical characteristic spectrum that indicates that a P450 is properly folded and has the heme prosthetic group. This is actually activity. So here we're seeing that there's a shift in the thermostability when we measure activity as well. But when we looked at resistance to hydrogen peroxide, we saw to our surprise that the oldest ancestor was considerably more resistant to higher concentrations of hydrogen peroxide as well. So if this is true for other ancestors, this gives us a way to solve that problem that we think limits the activity of P450s on long-term incubations. We can take advantage of the uncoupling of P450s, though, in a different way. So typically, as I was mentioning, P450s receive 2 single electron transfers. This is from NADPH via a redox partner, the P450 reductase. So here we have to have the P450 and the reductase, its redox partner, interacting to use NADPH. NADPH is an expensive cofactor, so it adds to the cost of biocatalysis. However, we can also take advantage of what's called the peroxide shunt. So oxygen surrogates, such as peroxides, organic peroxides, or hydrogen peroxide, and hypervalent iodine compounds can be used to short-circuit this reaction cycle, avoiding the need for either a redox partner or NADPH, or in fact oxygen. So they basically supply all of those components needed to get the P450 from the resting state to the active enzyme form that performs monooxygenation. So when we use this peroxide shunt, we can actually achieve good levels of activity with our ancestral forms. For example, again, looking at 6-beta-hydroxy testosterone production, we can see here the production of 6-beta-hydroxy testosterone by CYP3N1. This is at 2 different temperatures, 37 degrees in blue, 50 degrees in red. And you can see that we're getting almost quantitative turnover or quantitative conversion of testosterone that was present at 500 micromolar to its 6-beta metabolite. So this was run over a course of 12 hours. And this is supported by stepwise aliquots of cumene hydroperoxide in this case. So we're basically drip feeding the cumene hydroperoxide into the reactions. We did this with another enzyme, CYP2CE. This was with a different oxygen surrogate. This is a hypervalent iodine compound. And you can see that when we compare to the cognate system, the natural system using reductase, HCPR, this is abbreviation for reductase, and NADPH, by drip feeding BIIB instead, we get roughly a 40-fold increase in the yield. So this is sometimes quite marked in improving the yield that we can get from these enzymes. But of course, organic hydroperoxides are like hydrogen peroxide, and they can damage a P450, the protein and the heme. So we wanted to see whether our enzymes were stable to organic hydroperoxides. I should say that this experiment is not complete. We're in the process of going through our whole library of all of our thermostable enzymes to see their relative stability to cumene hydroperoxide. But I wanted to show the results of the first screening we did, which was on the CYP4 ancestors. So we can see here, these are the ancestral CYP4s here. These are the extant forms. Normally the human forms in 2 cases and a rabbit form for CYP4B1. Where we've got filled symbols, this is in the presence of cumene hydroperoxide, and the open symbols have no cumene hydroperoxide, sorry. So you can see that in almost all of these cases, there's not much of a difference between the amount of P450 in the presence and the absence of cumene hydroperoxide. And this is roughly 10 times the concentration we would normally use to supplement activity. However, there's one lineage that seems to be sensitive to cumene hydroperoxide. So we can actually get some information from this. We can look at the differences between these lineages and get some clues as to what to do to the P450 to make it more stable to cumene hydroperoxide. So in particular, these enzymes have a covalent attachment of the heme to the P450, except for the 4XZ lineage. So we think that that lineage is sensitive because it lacks this covalent attachment. And we think, therefore, that if we can add the covalent attachment to other forms, we can stabilize them to cumene hydroperoxide. So we're getting some clues as to how to further engineer these enzymes to be more stable under certain conditions. And I should say that we're pursuing this at the moment in a couple of co-supervised PhD projects between John, who's here online, and is another member of the hub. So John and I are supervising a student in each of our labs under a co-tutelle system. And these students are working on trying to improve the stability of these enzymes to oxidizing conditions particularly, and to scaling up. So to sort of summarize this part of the talk, we have a large library now of enzymes, ancestral enzymes, and also the corresponding extant forms. Here I'm showing the extant forms in plain text and the ancestral enzymes in bold. These come from all of the major drug metabolizing families, as well as some other families, CYP4s. And we can now use these to screen for particular activities. And I just want to show some results that we obtained screening against a few typical drugs for metabolite generation. So in all of these cases, these names are a little bit complicated. Basically, the stem of the name indicates the subfamily that the P450 has been inferred from. There's usually something that indicates whether it's from a particular phylogenetic group, and there's an N number, and the N number simply means the node of the evolutionary tree that it corresponds to. So it's a complicated name, but the names aren't typically important. We screened this library of enzymes against these drugs here, a couple of proprietary compounds, as well as these typical medications, all under the same conditions shown here. And I should say I'm going through these slides quite quickly. If you would like to drill down and have a look at some of the detail in these slides, they will be available after this webinar. So first of all, verapamil. So verapamil we found to be converted, like in human liver microsomes, to several dealkylation products. So there's, as shown here, there's a couple of O-demethylation reactions and N-demethylation, but there's also this N-dealkylation product. Also, there's some hydroxylations that we were able to see at very low turnovers. But you can see these are the major products that result from dealkylations. And each of these different enzymes has a different regioselectivity. So this, I hope, illustrates that if, for example, we wanted this particular metabolite at 4.04 minutes in the elution, we'd use this enzyme to produce it. If we wanted the one at 4.29, we'd use this enzyme. So we can choose the enzymes to use for metabolite generation because they all have slightly different regioselectivities. We can also see here that we've got good activities in many cases with the oxygen surrogate. So this is these are the enzymes that were the best at each of those particular metabolite productions. And this is just looking at the raw yields from that incubation. So even without any optimization, we're getting reasonable yields of these metabolites. This is apixaban. There are a lot of different metabolites of apixaban, at least 8 hydroxylated metabolites. Most of them produced at relatively low yields. You can see that different forms produced different metabolites as their major metabolites. So this shows the best enzyme for producing particular metabolites. So looking at 4 forms here, we've got 2 different excellent forms and a couple of ancestors here. You can see that they have quite different regioselectivity. We can also see that sometimes the oxygen surrogate can change the regioselectivity. So here we're getting better yields of certain metabolites when we use an oxygen surrogate compared to when we use the NADPH and reductase to support activity. This is one of the proprietary compounds, and I show this because it's a sort of typical result. We've got a lot of different extant forms here and the corresponding ancestral forms. So here we basically found metabolites from N and O dealkylation and aliphatic and benzylic hydroxylation. I can't show the structure of this compound, but those were the types of reactions that were catalyzed. Basically, the ancestral P450s recapitulated the pathways that were found with the human P450s and in human liver microsomes. The ancestors tended to metabolize or tended to catalyze the same reactions as the extant forms in the same subfamily. However, we did have some novel products. So this product was completely novel, was only found with this particular CYP3N19 enzyme. This enzyme, this product rather, would only have been found in this experiment, given that the only other extant form that produces it is not one of the big 5, not typically one of the forms that would be used for screening. This is actually a goat enzyme. So this wouldn't have been found in any typical DMPK analyses. And sometimes the ancestors showed altered regioselectivity, or minor metabolites were more prominent in some cases. And finally, I wanted to talk about one particular proprietary compound. We use this as an example of an assay to produce milligram quantities of a particular metabolite, say a compound that was flagged as of importance in missed considerations. So in this case, this compound Y was transformed to one major hydroxylated metabolite and one minor hydroxylated metabolite. And this was catalyzed by CYP3 forms exclusively, both the major and the minor metabolites. And CYP3N19 was the best of these forms. So we basically produced a lot of CYP3N19. We produced a large-scale culture, but a very straightforward large-scale culture. We produced bacterial membranes. So this, I should have said this before, typically we use these enzymes in a crude enzyme preparation form. They're the membranes isolated from E. coli that are expressing these P450s. Effectively, we can use these just like we do human liver microsomes or animal liver microsomes generally. So we obtained, we did a large-scale culture, a 1 liter culture of this enzyme, isolated the P450, and performed 2 600 ml reactions in shake flasks with bacterial membranes, 0.5 micromolar P450 with reductase, and 75 micromolar of this compound, which ended up being, across the 2 reactions, about 40 milligrams. We had a modest turnover, not a great turnover, only 24%. Combined reaction extract, we estimated we had 9 milligrams of product. We lost quite a bit in the workup because we're not a lab that's set up for preparative scale compound isolation. But at the end of this very simple experiment, we came up with 5.6 milligrams. And this was in very pure form, as shown here. So this is the crude reaction extract. This is a purified metabolite. and this was a weighed amount. So this was enough to establish the structure by NMR and also to do some preliminary toxicology assays. So we think this gives a good example of how in very short order, very quickly, we can generate metabolites. So this wouldn't have taken more than probably about 6 working days to screen and do the preparative analysis. So in the last part of the talk, I'd just like to illustrate some of the ways in which we're developing these enzymes for as basically biobricks for synthetic biology. So there's the ability to stabilize these enzymes allows us to do a lot of other things to them, which has really opened up a lot of possibilities. I've already talked about the monooxygenase chemistry without oxygen, but I wanted to address a few different ways in which we can use these enzymes. So first of all, non-natural chemistry. So you'll probably be aware of the work by Frances Arnold and her lab, where they've been looking at catalyzing non-natural reactions using P450s and other heme proteins. So this is a classic example of cyclopropionation of styrene. So to do this, Frances's lab modified the cysteine thiolate ligand to the heme, the normal cysteine that holds the heme in place. They had to do this to get good activity, and they modified it to a serine and a histidine and a couple of other alternatives. And they looked for the production of the cis cyclopropionation product. So we did this with some of our ancestors, and you can see here, compared to the control cells, this is actually in whole cells. The whole cells with the hemoproteins that are in E. coli, we can get some of the trans product, but it's only when we have one of these engineered P450s, or P411s or P422s, as they're called, because the absorbance spectrum changes. So these enzymes will produce the cis isomer. So we were heartened to see that our enzymes could do what the engineered BM3 enzymes could do. However, we also went back to look at the P450s themselves without that modification of the cysteine, which I should say destabilizes the enzyme a little bit. And when we did this, we actually found a P450 that didn't have this mutation that was much, much better than any of the P411 or P422 forms. So you can see here from the break in the axis, this particular P450, the 2BS form, was particularly good at the cis cyclopropionation of styrene. So we would contend that it's not actually necessary to modify the conserved cysteine. We just need to pick the right P450. Moving on to another application, we've been interested in exploring the immobilization of P450s on protein scaffolds, so nanobioreactors, as we call them. So in particular, we've looked at virus-like particles. So these basically are the capsid of a virus particle. So we don't have any of the viral DNA. We just have the proteins that make up the protein case of the virus. And we can express the P450 and the reductase as cargo on some of the proteins that make up this capsid. So in this case, we've got the P450 and the reductase on different proteins, and they basically self-assemble in host cells, in E. coli in this case, to produce virus-like particles that have a P450 and reductase inside the viral capsid. And this is a porous structure, and so substrate and cofactor can get in and out of the virus particle. So the idea here is that we would use these particles as the biocatalyst, produce them in E. coli, isolate them in a very straightforward fashion by differential centrifugation, and use them in incubations. The advantage here is that it's a lot easier to obtain something that's a large particle and pull it out of the reaction mixture than if you just have a protein. So it means that the reaction mixtures can be clarified to improve product recovery. It's particularly easy with certain viruses. This is a virus called BTV, blue tongue virus. This is actually produced in a plant culture system. But in this case, to get the virus particle to separate, we just need to drop the temperature to 4 degrees. So this means that just by simply dropping the temperature, we can separate out the catalyst, and we have a supernatant that is a nice clean mixture and will allow much easier product recovery. So we've done this now with a couple of different viruses, as I mentioned. I'm just going to show the results from a virus called P22. So here we're expressing both the P450 and the reductase on a scaffold protein. We co-express these scaffold proteins using a 3 plasmid system in E. coli. The third plasmid has the coat protein. So we need a coat protein and a scaffold protein for the virus-like particles to come together. You can see here that we've got some nice virus-like particles here. This is from that simple differential centrifugation step. So the microscopy shows that we've got a very, very nice clean mixture of particles. They're well formed and they're basically easy to separate. This shows some results from size exclusion chromatography. So here we're seeing a major peak that corresponds to the virus-like particle. There's also a little bit of an aggregate sometimes, but here looking at the reductase concentration, we can see this is based on reductase activity, and that lines up beautifully with the VLP peak. So we're actually getting the reductase in the virus-like particles. And likewise, we've got a P450 peak that again lines up with a major VLP peak. So we've got both the P450 and the reductase active or properly folded in these virus-like particles. We're just at the moment looking for activity from these co-expression virus-like particles, but we've looked at the activity so far with oxygen surrogates in particles that just contain the P450. And basically we see for all of these different P450s with different oxygen surrogates, we see that the activity with the virus-like particles is similar to the activity we see in bacterial membranes on a per P450 basis. So with equivalent amounts of P450, we see similar levels of activity. Finally, the third synthetic biology application I wanted to talk about is one that's quite exciting in terms of thinking about sustainability, true green chemistry. We have followed the lead of some researchers in Copenhagen with whom we've been collaborating to put P450s on the end of photosynthesis. So this shows the typical photosynthetic electron transport chain. It starts with the splitting of water to produce oxygen. Electrons are transferred through photosystems to a ferredoxin and then to a ferredoxin reductase, and they are used to reduce NADP to NADPH, which is used as some of the energy currency in cells. We can put a P450 on the end of this electron transport chain and use some of the excess electron flow through ferredoxin to supply the electrons for the P450. So here we're basically taking some of the electrons from photosynthesis, also the oxygen generated in photosynthesis, and using it to support a P450 reaction. We've been doing this work with Ben Hankemer, who's another investigator in the hub, and in Chlamydomonas reinhardtii, which is a microalga. This is a very useful organism because it's cheap to cultivate at various scales. It basically will grow on solar energy, on carbon dioxide and water. And you can see we can go from the micro scale where we can screen things very easily to larger and larger bioreactors to these open ponds. It's also useful because genetic manipulation of clammy is very well established. It's a lab organism and we can use tools such as CRISPR to modify its genome and to get the enzymes expressed in the chloroplast. It's also easy to do metabolic engineering, for example, to produce the precursors of our desired compound. So this is some in vitro results from a PhD student that Ben and I co-supervised. And here we're comparing this light-driven system where we're simply adding a P450, purified P450 to thylakoid membranes. And in the presence of light, looking for activity. We're comparing that to the typical system of P450s in bacterial membranes. So this is supporting activity using NADPH and the CPR. And this is a bit of a hybrid system where we're using that photosynthetic ferredoxin reductase and the ferredoxin using NADPH, but running the system backwards so that we're getting the electrons from NADPH through the ferredoxin onto the P450. So when we compare the activity with these three different systems, we see that this is the natural system with the reductase supported by the NADPH. that's not as effective as the light-driven system. The light-driven system is more effective at producing, in this case, hydroxylated diclofenac metabolite. And this intermediate system is the worst of the three. When we look at the amount of diclofenac turnover, we can see we get almost 70% turnover within 3 hours of incubation. And we're really not seeing much of a loss of activity over that time. It's going to equilibrium, but we're not really seeing the system die off. And we've also shown that this can be done in vivo, in clammy, so we can express these enzymes. We've now got several P450s expressed in clammy. You can see this is a Western blot showing the P450. This is another P450. And we can show light-dependent activity in clammy. Clammy itself has some activity towards certain drug substrates, such as diclofenac and testosterone, but we've got a clear difference in the presence of the enzyme as well. So this is particularly exciting, we think, for that last part of this pipeline here, the waste stream bioremediation. And this is something that's often ignored, or it's a bit of a problem. But we could see, for example, algae transformed with particular P450s in containment ponds at the end of a drug pipeline, a production pipeline, being used to treat the waste from that pipeline before it's released into the environment. So basically, we think that we've really addressed a lot of the issues with regard to using P450 enzymes in applications in industry. Having got these results, it started a conversation about what we can do now to get the enzymes used to their full potential in industry. So what to now, where to now, and how to get there. So about a year and a half ago, we started talking as a group about what we should do, how we could make use of this and really achieve a bit of a step change in the use of enzymes in industry. Because P450s generally haven't been used to their full potential. Other enzymes have, but not P450s, and also not several other enzymes like UPOs, halogenases, alkyl transferases. So building on our platform technology of being able to stabilize enzymes, not just P450s, but other enzymes as well, we've consulted widely with industry to try and determine what the key problems are that need to be solved. We've assembled a critical mass of academic investigators with complementary skills that are relevant to biocatalysis. And we're at the moment trying to bring together industry partners in the pre-competitive space to design projects that address the needs of industry. So our vision is to have an academic industry consortium to unlock the potential of enzymes for use in pharma by addressing the factors that limit the scale-up and cost-effectiveness of biocatalysis. So our aim here with this hub is to leverage funding from the Australian government for an ARC industrial transformation research hub in biocatalysis for pharma. If we can get this funding, we hope to engage, or we will engage, teams of researchers on projects that are designed with industry partners. And we hope in this to achieve a step change in the implementation of enzymes in industry. So our discussions so far, to which the companies, many of the companies represented here, have contributed very substantially. They've identified these priorities. First of all, the scale-up of enzyme reactions. So applications beyond metabolite generation. We can do metabolite generation, as I hope I've demonstrated, but applications at larger scale still need a bit of work to work out how to effectively scale up so that the yields scale. There's also interest in novel chemistries, so nitrogen transfer, carbon silicon bond formation, those the other catalogue of reactions that the Arnold lab has found to be possible with hemoproteins. Halogenation is a particular a reaction of particular importance, particularly fluorination. And we'd like to find some halogenases or fluorinases particularly that we can stabilise and implement in industry. So stabilisation of other enzymes, UPOs, halogenases, alkyl transferases, also finding novel enzymes from metagenomes. We want to find out how we can best implement these enzymes into processes, such as in reaction cascades, but also blending with other methods of catalysis, chemo and photocatalysis. Also immobilizing enzymes in catalysis in flow has been pointed out as a priority. But finally, the last domain is really deciding which enzymes to use and when, where, how, and why. Working out which enzymes to screen first, which enzymes to prioritize. There's a lot of different enzymes available for metabolite generation. Where do you start? How do you cut your screening costs? By using the most likely enzymes first. Also, decision trees for biocatalysis, working out what system to do the biocatalysis in, whole cells, purified enzyme, that sort of decision. Also, how to work out how to combine most effectively chemocatalysis and biocatalysis. Techno-economic analysis of processes to work out the best process design, and life cycle analysis to achieve improvements in sustainability. But we're also interested in other suggestions. So if you have any other priorities that would be good for us to address, we're very interested to hear your ideas and very interested to incorporate these into the plan for the hub. So the value proposition for industry partners wanting to get involved in this hub, we are going to be providing opportunities to design projects that solve problems that are not easily addressed within the constraints of industry R&D programs. We will provide access to internationally prominent researchers with complementary expertise in specialist relevant areas. We can also strengthen the talent pipeline for industry because we'll be training up some excellent graduates in these specialist skills. And there's opportunities for co-supervision, internship, and industry visits. I apologize, I'm losing my voice because I'm just recovering from a respiratory infection. And I've been talking for a little bit too long. We also will be providing simplified access to our platform technology of thermostable enzymes. Early access to relevant novel technology and tools, including databases of biocatalysts, software for priority screening, and techno-economic models. And we're going to be communicating the results of our research through monthly research updates with partners for each individual project, but also 6 monthly online forums to basically communicate, to disseminate the results of the hub as a whole. And these will be recorded and accessible on demand. Our timeline for securing the funding from the government, we're working up to a deadline in late November. The 26th of November is when we have to have our application in. So we're really trying to secure participation of industry up to that point. I should mention that at that point, we only need non-binding letters of intent from potential partners to say, we are interested in this project. We have the intention to contribute in whatever way is reasonable. In terms of funding, because there's time taken for the grant to be reviewed, we'll find out whether the results, what the results of the application are mid next year, so mid 2025. We are likely to hear whether we get the grant or not. And there's a year that's allowed for negotiation of contracts after that. So it's only when those contracts are signed that the agreements become legally binding. And so we anticipate that the actual research will only start mid-2026 and it will extend for 5 years. So in terms of budget timelines, the funding is only needed in the second half of 2026 for the 5 years following that. And before I finish, I need to thank the people who have actually done the work here. So on this slide, on the left-hand side, these are people from my lab. On the right-hand side, we've had a number of excellent collaborators. I've mentioned a couple of them already, Ben Hankemer and Frank Sainsbury, but these are also other collaborators that we've worked with. Birwa Lindberg-Moller, was our collaborator for the photosynthetic work as well as Ben, and Emily has been involved in the crystallization work. I would like to thank all of these participants, but also I'd like to thank both the Australian government for funding, but also AstraZeneca for funding a lot of this work. And I'll leave you on this slide. So this is basically our vision for the objectives that we're working towards for the hub, and it reiterates the structure that we have with this complementary expertise. So I'll close now and take some questions, and I might just stop sharing so that we can see some faces. So thank you for your attention, and I'd be very happy to entertain any questions that you might have.